So today's video is relating to the liver, as you can see on screen at the moment, and this is part of one of the excretion topics that we look at. So with regards to the liver, this is a really important organ to the human excretion system. So the liver is actually involved in over 200 different processes within the body, so it's an extremely important organ. You don't need to know all of these when we're looking at it from the point of view of A level, but there are certain things that we should be aware of. So probably one of the first things to say is that in your mammal, the liver is concerned with the conversion of ammonia to urea, and that has a huge draw in terms of water. So when we're looking at the water uses, there's approximately 95% water used to dilute this toxic product, and the ornithine cycle is used to convert the ammonia into urea. So we start by looking at the liver as a whole then. So the liver is made up of hepatocytes, and if we look at those hepatocytes, there's approximately 300 billion of these, so there is a lot of these cells. Now, looking at these cells, if we say we've got approximately 300 billion of these, and these are incredibly metabolically active, so high metabolic rate, and that means that they can divide and replicate, which is really important to the role of the liver. So here we need to also note that with regards to these cells, in order to support them being metabolically active, they have got a large nucleus, prominent Golgi body, and they've also got a significant amount of endoplasmic reticulum in order to support this metabolic activity. Now, if we're looking at this and this high metabolic rate, in actual fact, your liver can regenerate in 90 days. So it can completely regenerate in approximately 90 days for this regeneration. Now you can donate up to 2/3 of your liver. Okay, so that means about 65% of it can be donated, and your peak synthesis for this is going to be 7 to 10 days post-surgery. So 7 to 10 days is your peak synthesis for this regeneration. And that's really important. So when we go on to look at the liver, its ability to regenerate and restore is one of the things that means that actually in some cases people abuse their liver because they understand that it has this ability to regenerate. These cells can renew themselves, but when it comes to things such as consumption of alcohol and consumption of drugs, sometimes this is a key element of the liver that ends up being abused because of that. Now, interestingly, we have known about this regeneration actually since the ancient Greeks. So if we look at the ancient Greek mythology, actually we see the story of the punishment by the gods of Prometheus. And so what they do in this story, you see him being chained and the birds come down where he's chained to this rock, and they actually, the vultures would peck out his liver every day. Yet overnight we see the regeneration of this. So we understand from this that actually we've known about this since the ancient Greek times. So this is something that we have had this awareness of for a significant time. So the next thing to note about the liver is its weight. So if we're looking at the weight of the liver, we're looking at approximately 1.3 kg for a female. And then we're looking at approximately 1.8 kg for your male. Okay, and so we do see a slight difference between the average weight between the genders. We can also start to note its wider structure past the hepatocytes. Okay, so in terms of the structure, we need to note that this is a lobed organ, and that those lobes are divided into lobules, and that those lobules are made up of the hepatocytes. And again, we note here that there's approximately 3 billion of those. Okay, so approximately 300 billion. So that's very important to this. Okay, the liver is also important to recognise the blood supply. Now, this is an organ that takes a significant amount of blood that is branching off from the main aorta, and we have got approximately 13% of the body's volume of blood can be held within the liver. Now, as you've got a significant amount of blood being held here, that means that actually if there was an accident, you can release some of this blood, and in releasing the blood, that's going to enable you to increase the blood pressure as a result of that. Okay, so that can be a significant advantage, but obviously it can only be released once. In terms of the amount of blood that's passing through and being filtered, you've got approximately 1.7 litres per minute, which is quite a significant amount. Okay, and the blood which is coming into and out of this organ is coming through what is referred to as your hepatic vessels, and we'll actually look at the different hepatic vessels which are coming in. So you have 3 vessels that you need to be aware of in total, and we will look at how you can identify those vessels and the different blood sources, where those are coming from and what is contained within those vessels. So if we look now at the structure of the liver, you can see the liver we've just looked at, and we can see here that this is divided, as we've said already, into lobes. So you've got sort of 4 main lobes that make up the liver, and those lobes are divided into your lobules, and you can see here these hexagonal structures. Now, your lobule is your basic structural and functional unit of the liver, and each one of these, what looks like hexagons sort of interconnected here, is a lobule. So we'll just label this quickly. So you've got our liver, which is lobed, and then we have our lobules, which is the functional and structural unit. And here we have an individual lobule. Now, as we can see here, this lobule itself is made up of a number of different structures. So actually we can see that you have got the hepatocytes, and that these hepatocytes are surrounded by a number of vessels. So we have our hepatic venule in the middle, and we then have our branches of our hepatic artery, which we can see in red. We have the bile duct in green, And we will look at these, we will look at the caniculi in as well in relation to these. And then we also have the hepatic portal vein that we need to be recognising too. So we've got our hepatic. And what we need to do is recognise how these are working together and how this lobule works and the different components of this. So just to touch briefly before we look at the detail and sort of go even sort of further in and look at a sinusoid here. So we've said that the lobules are the basic functional and structural unit of your liver. If we look at your hepatic venule, this is acting as a sort of almost like a drain, and this will drain sort of all of the blood inside of the lobule. The hepatic artery are organised around the outside, and they're bringing oxygenated blood into the lobule and into the liver. And we then have your hepatic portal vein. Now that is carrying what would technically be referred to as deoxygenated blood, but actually does have a significant amount of oxygen still within it. This is coming from the intestines. So the importance of this particular vessel is that it's going to be bringing nutrients to the liver and to the lobules. So the final structure then on here that we need to look at before we delve deeper would be your bile caniculus. Now this is approximately 1 to 2 micrometers wide. And these are tissue spaces between the membrane of the adjacent hepatocytes. Now that means that they're actually completely sort of encapsulated, and we'll see this when we zoom in and sort of look at this in greater depth. And so this creates then the sort of interlobular network of channels, and those drain the bile that's produced by the hepatocytes, and that will then drain down into the bile ducts, and those bile ducts run sort of around the outside. So we actually need to sort of improve upon our labels, because where I've labelled that at the moment is technically going to the caniculus, whereas the bile duct would be running around sort of the outside of this. So let's look in detail then. Let's start to look at what the sinusoid looks like, the interactions of the blood within that sinusoid, where the caniculus is, and how the cells are sort of interspersed and supported around this. Okay, so let's just look in detail then into one of the sinusoids of the lobule that we had been looking at, and we can start to just put those labels back on that we were looking at. Okay, so let's start with the blood that is entering the sinusoid. So if we start here, first of all, we've got our hepatic artery. And if we're looking at that now, what we need to recognise here is this is going to have a thicker wall because of it is an artery. So we'll try and represent that here. And it's also going to have a smaller lumen. So we can look for thick wall and small lumen. And we can see that the blood is going to enter here, and it's going to enter into this space. That was a little bit thick, that arrow there, and that the blood is going to be moving up into this space here. So we will see that oxygenated blood which is coming through here, and we would see this sort of moving all sort of into this space. Okay, then when we look at our next area in terms of blood which is entering, we can see that we also have blood which is entering via your hepatic portal vein. And this is technically deoxygenated, but we do know that there is actually quite a significant amount of oxygen that is here. But the importance of this is this is going to bring blood which is coming from the intestines to the liver. It has a thinner wall and it has a larger lumen. So I'm just going to put from intestines here, and we can recognise that. So we're going to colour this blue, even though it does have some oxygen within there still, and we can start to see that actually there will be a mixing of the blood within this space. So we'll actually see a mixing of both the oxygenated and the deoxygenated blood which is coming through. So that would mix within this space. And so we have both the oxygen being provided to the hepatocytes, but also the glucose, which is coming directly from the products of digestion. The next thing to note is the caniculus, the bile caniculus. So here we have the this space which is completely encapsulated by the hepatocytes, and that is important because that means that being surrounded in this way, that bile can drain, and it can drain down into the bile duct and then ultimately go to your gallbladder. Now this is quite an important thing to note here. So you're going to have the bile caniculus, they're going to drain into the bile duct, and you've actually sort of got this biliary tree, so this sort of network of interconnecting vessels that will ultimately all end up draining into the gallbladder. Equally, if we look here, at the moment we have had a situation where we have had two vessels with blood entering into this sinusoid. However, we've now got the bile draining in the opposing direction away from the hepatic venule. So we can just add some arrows on here just to represent this. So I'm going to put them in black so they stand out. Everything else is going to be moving from the sort of outside of the hexagon, if we were looking at the whole lobule, and draining into that hepatic venule, which is at the centre of that lobule. So the very last part of this I wanted to mention to you was the cell that we haven't yet recognised, and that is a Kupffer cell. So this is a phagocytic cell which is found within the lobules and within the liver tissue, and these play a really important role as a first line of defence in being able to destroy anything and engulf anything that should be found within the blood that is passing through this space. So the final thing to mention is the role of the hepatocytes, and they've really got sort of four key things that they are involved within, three of which are homeostatic roles of the liver. So the first of the things that we need to consider is that they make bile, and this emulsifies fats, and then that increases surface area for the digestion of those fats, so that increases the rate of their digestion, and it needs to be noted this is actually sort of a physical and not chemical breakdown, the emulsification. They're also involved in glucoregulation, so they help with the conversion of glucose to glycogen, and we see glucogenesis here. That regulates your blood sugar levels, and they also respond to insulin and to glucagon, so they've got receptors that are upon them that bind to and respond to glucose in this way. They're involved in deamination, so in relation to this you need to check back in with the ornithine cycle, and this involves removal of the amine group of the amino acids. The amine group will then become urea, so be converted from NH2 to NH3, and that is using the ornithine cycle, and then sent to the kidney for removal, whereas the rest of that amino acid can become pyruvate, and that can actually be then used in respiration in order to generate ATP. Now this can be used when there's not enough glucose for respiration, so the body can break down the protein to release that pyruvate. However, it would result in a loss of muscle mass. The final role that we have noted here is detoxification. So the liver plays an important role in the breakdown of alcohol and drugs, but also processing any ingested toxins. So with regards to alcohol, the liver has an enzyme called alcohol dehydrogenase, and that converts firstly ethanol to ethanal and then to ethanoate, and then the ethanoate can be converted into fatty acids, which can be used for storage and respiration. So just finally, I wanted to go back up to the Kupffer cell, which we mentioned was phagocytic. Now, not only does the Kupffer cell play an important role in being a first line of defence in engulfing any sort of bacterium or any pathogen that was found within the blood passing through the liver, but also plays an important role in breaking down any old or senescent red blood cells, and that then actually releases the heme, which breaks down into biliverdin and also into iron. Now, the biliverdin actually then gives your colour to your faeces, and the iron can then be either stored or it can be used, and in the female it can actually be lost through menstruation, whereas the globin part of the hemoglobin can then be hydrolyzed to 3 amino acids. Okay, I hope that has been helpful. So that just summarises the liver, the structure of the liver, and the functional unit of the liver, the lobule and the cells which make that up, the hepatocytes, and so just a very brief overview of the roles and the many roles played. We said up to 200 different functions that the liver is involved in overall, so certainly some wider reading to be had in relation to those roles.